Let's continue with our introductory talk on physiology. Do you guys have any questions from the Tuesday lecture?
Oh yeah, we didn't finish. We're still on the first one.
We're still on the first one. Yeah, we got to finish that up, and then we're gonna just kind of breeze right into the second lecture, which should be posted. Yes, ma'am.
So you know how you were talking about you can become your baroreceptors can become like insensitive to hypertension.
Yeah.
Is that the same case for hyperglycemia?
In some cases, that can be the case. So what you end up finding for, say, like type 2 diabetics, right? So say there's a person who have, sedentary lifestyle, they have poor dietary intake, and they're just kind of constantly kind of riding high on their glucose, right? So what happens with diabetics, which you'll learn in other courses, obviously in pharma and whatnot, is that their pancreas is kind of at, they're kind of in overdrive a little bit, right? So they're secreting too much insulin in a lot of those cases. So sometimes what happens for them is when they get their receptors on all of the other cells peripherally that normally respond well to insulin to intake glucose and other nutrients, they get insensitive there, right? So you see this downregulation where there are fewer receptors for the insulin to respond to, and so the pancreas is trying to respond because it doesn't have that negative feedback loop, right? Because the blood sugar is still high, so it keeps sending out more and more insulin, right? So we call that insulin resistance, and so you see that in a lot of peripheral tissues. The other thing that you can see is that the pancreas, the beta cells themselves may become worn out over time. So you're just constantly working them too hard. They say, you know what, I'm done. I'm going to quit early, say like in their 50s versus like they would maybe if they had better diet and things like that, and maybe it would wear out in the 70s or something, right? So it's just kind of an enhanced degradation of those type cells for spitting out insulin. So we'll have patients who, for instance, like if you were to start like a new onset type 1 diabetic versus a new onset type 2 diabetic, if you look at their insulin doses, they vary wildly, right? So for instance, type 1 diabetic, they just ran out of insulin because their beta cells were destroyed, right? So they do not have any insulin resistance, so they can have very small doses of insulin. So say like 3 units of insulin is enough to get them corrected versus some of the type 2 diabetics, they may need 8, 10 units of insulin to get the same kind of effect because their cells are just very insensitive to that insulin. Yep, so that would be another case where you'd have that kind of alteration of what the normal set point would be. Yep. Yes, ma'am.
For the intercalated discs in the cardiac muscle, all I really think I got from that is they allow the passage of sodium ions between cells so the entire heart doesn't contract at once. Is that just like the atria and the ventricles?
Yeah, so we'll talk more about this when we get to the actual cardiac physiology section, but essentially, where does a heartbeat normally start? Yeah, so SA node, right? So that's that sinoatrial node. That's where the initial heartbeat starts. That's where your initial action potential. We'll learn that there's pacemaker cells that kind of can initiate action potentials on their own, and then there's other cells which are not pacemaker cells, and they rely on that input of an action potential to start up, right? And so the idea is you start in the atria, you travel down through the ventricles, and then you get this nice kind of concerted contraction of the atria and then the ventricles, and you get blood flow, right? If you did not have that ability for those action potentials to travel so nicely in a kind of an organized fashion, you'd end up having arrhythmias happen, right? So maybe the atria would be beating too fast and it wouldn't be communicating with the ventricles very well. Sometimes you'll have like accessory pathways where tissue will feed back into another area, and that's where you can have things like Wolff-Parkinson-White can happen, different things like that where you can have these arrhythmias pop up due to altered conduction pathways. But again, we'll talk more about that when we get to the cardiac section.
Okay.
Any other questions? Fantastic. All right, so we're going to continue on. We're talking about different types of chemicals and different substances within the body that are useful for living, right? So another thing we'll talk about are going to be prostaglandins. This is really important, especially when we get to the pharmacology sections, because we have a lot of medications that can deal with prostaglandins. Basically, it's a type of fatty acid. A lot of the prostaglandins that we have are actually derived from some of the membrane phospholipids that we talked about last time in the cell membrane. So some of those, we'll talk about the arachidonic acid pathway and how that can be taken from some of those lipid, the lipid bilayer. You can produce prostaglandins from that. So prostaglandins are very, very important. They help to do things like regulate our blood pressure. They also help to regulate blood flow to different organs. So say, for instance, in the kidneys, it's really important that we have prostaglandins because they, in a lot of cases, can be vasodilatory, right? So it's going to cause vasorelaxation and cause increased blood flow to certain organs. So we talk about kidney blood flow. We'll talk about the prostaglandins being important for blood coming into the kidneys versus blood coming out of the kidneys, right? And so we'll also see that certain prostaglandins are useful for things like ovulation and helping with uterine contractions. In some cases, we'll actually give certain types of prostaglandins to women who need help with that, getting their cervix ripened so they can actually induce labor. So you might actually see that for someone who's having kind of like delayed labor. It's also really important for inflammation. So anytime you have inflammation that's occurring, say you cut yourself or you're spraining an ankle or you have a headache, prostaglandins are usually going to be used there in order to help kind of stimulate that process. It'll cause vasodilation, you have increased blood flow, that's where you get a lot of that warmth, that's where you get a lot of that redness that comes about with some of that inflammation that happens there. Can you guys think of any medications that might affect this kind of inflammation from prostaglandins? Probably take it every time you have a headache. Yeah, NSAIDs are going to be a big one that help to blunt the production of prostaglandins by inhibiting some enzymes. So things like ibuprofen, things like aspirin, things like naproxen, they actually work through inhibiting the system. So you can see how they can work as anti-inflammatories by dealing with these substances that are pro-inflammatory. Also useful for blood clotting as well, which you'll learn that one of the problems with NSAIDs is that you see an increased risk for bleeding. So again, if you interact with this process, you can not only have the therapeutic effects, but also see some toxic effects as well, just due to the fact that these prostaglandins are being kind of inhibited kind of in a blanket fashion. You can see here kind of their basic chemical structure. Notice they are basically the fatty acids where you have basically the, you can see how they've kind of been developed off of fatty acids and maybe been broken off of like, triglycerols and things like that, and so they can be lots of different varieties of prostaglandins. Some of them you'll see are important for things like angiogenesis. They're important for producing new blood vessels to make sure we're delivering oxygen to the right tissues. These are also really important for helping with the gastric barrier, right? Your stomach produces a lot of acids, and if you didn't have any sort of mucus barrier there, you would actually just bore a hole right through the stomach. And so prostaglandins are useful for helping to produce that kind of mucus layer that helps to protect the stomach from its own acids. So we'll talk much more about these, especially in the pharm section. But just know that they're really important for blood vessel dilation and constriction, important for blood clotting, important for inflammation. Lots of different things we see prostaglandins being used for. All right, moving on, we'll talk about our proteins. And so, again, these are going to be composed of an amino acid chain. We'll look and see some of the basic structure of those in just a second. There are 20 different main amino acids. When I was taking biochem in pharm school, I had to memorize all of those. I'm not going to make you guys do that because that would be mean. But just be aware that there are lots of different varieties. And so by combining these different amino acids together, you get different proteins, right? And so based on their structure and how they kind of fold and how they kind of come together, you can get lots of different structures. You can get transporters, you can get enzymes, all kinds of different things from these amino acids. And so that structure, that sequence of amino acids is going to be dictated by DNA, right? DNA is going to code for some of these different proteins. Do you know the main molecule that will use that DNA to form the proteins? Yeah, so the ribosomes, but they use so we'll use RNA basically. I will talk more about that in detail, but use RNA to help to transcribe and translate that information over to help make these proteins, right? So we'll talk about that process in more detail. You can notice here where we get the amino acid name from. You can see right here you have the amino group, and then you have the acid group, and then the only thing that really differentiates them is going to be their functional groups. Anytime you see R, that's basically just a functional group that can differ between the different proteins within the class, right? So say for instance, you have valine, you have here just kind of trimethyl structure here in tyrosine. Tyrosine is really, really important. You'll see that that's really important for things like neurotransmitter function. It actually produces things like dopamine and norepinephrine, so that'll come up very frequently, especially when we talk about psych drugs later on in the year. And then again, by connecting these different proteins together, or amino acids together, you get the proteins that are going to be functional in nature.
Are we expected to know the structures?
No. No, I had to be able to draw structures. I don't think you guys need to do that. Yeah, no problem. I'll be mean elsewhere, but this will not be the place that I mean. Yeah, so, yes, ma'am.
Does that go for the structures throughout this whole PowerPoint too, or just the amino acids?
Yes, I'm not going to like put a picture of a chemical structure and be like, hey, what is this?
Okay, so like the one up with like the prostaglandins here, like with the
those are just examples to show you that, hey, they're fatty acids. You notice how they have like a polar and a non-polar group. And you notice there, when you're looking at them, they look kind of like those phospholipids that we were looking at before. Notice how one part's polar, one part's non-polar, and that can kind of help getting it to certain places. You'll find that it helps to deal with solubility and things like that. But basically, they're just derivatives of fatty acids. Yep. Again, you can see how you can connect these together. Basically, the amino group connects to the carboxylic acid group, and they're able to form these chains, and thus you get your proteins, right? Do you guys think of any like important proteins in the body that you may rely on?
Enzymes.
Enzymes, yep. So enzymes are going to be super important. Hemoglobin, yep. Just going to see some hemoglobin, some proteins are going to be involved there. Insulin's going to be another big one. We've been talking about that pretty extensively. So lots of proteins everywhere that we are very reliant on in order to live. You can conjugate proteins with other substances as well. So you can have proteins being conjugated with carbohydrates. Those would be called glycoproteins. Basically just a carbohydrate plus a protein. You'll find that these are important for certain hormones. You'll also find a lot of them being stuck into cell membranes. Some of these can be useful for either identifying a cell, either as self or as other. You'll find it being useful for certain receptors and things like that. So those can be very useful. You can see a picture here where you can see some of these glycoproteins kind of being embedded within that phospholipid bilayer, that cell membrane, and they're able to interact with other molecules, right? And then you also have lipoproteins, basically just a protein plus a lipid. You find these also, again, in cell membranes, but also they can be used as carrier molecules as well. Like lipoprotein lipase is a very important one for like cholesterol metabolism, and so you'll see that being used for when you talk about things like production of LDL, which is good or bad cholesterol. You guys know? Yeah, LDL is like the bad cholesterol, and then you have like HDL, which is like the good cholesterol, and a lot of that is related back to lipoproteins and being able to process those. So just some examples of other conjugated proteins, like I already mentioned, hemoglobin is really important. What does hemoglobin do for us? Yep, so it's going to transport oxygen to different areas in the body, which is very useful, right? We have myoglobin. What is that going to do? Yep, so we're going to have oxygen in the muscles, which is really important because oftentimes we can run out of myoglobin, or not run out of the myoglobin, we run out of the oxygen carried by the myoglobin, which is why we have a lot of anaerobic metabolism that goes on in the muscles. We'll talk more about that in a few lectures. Insulin is really important. We already mentioned that as regulating blood sugar, usually driving it down and driving blood sugar into the cells. And you also have like your blood group proteins. What does this do? Do you think? Yeah, so what are like some different blood types? Yeah, so A, B, O, A, B. All right, so those are coded by some of these glycoproteins. So basically by having the presence of those glycoproteins, you get typed as having a specific type of blood. Yes?
If insulin doesn't have a non-protein component, then how is it conjugated protein?
Basically, because it comes out as actually you can have two proteins actually being linked together. That's a good question. So later on when we talk about the endocrine system, you'll actually find that insulin is actually two components when it comes out initially from the beta cells. It's proinsulin. So that proinsulin actually has a C protein that's conjugated to the actual insulin molecule itself, and that can actually be useful. You can actually tell if someone is having like a hypersecretory condition. If they have like an insulinoma, you can measure high levels of that C protein, and that'll actually give you an idea if they're secreting too much insulin or not. But yeah, so that would be one where proteins conjugated to another protein, and then it's an inactive form. When you cleave that, it actually comes out as active insulin. Very good question. Thank you for pointing that out. Yeah, so the blood groups are very useful because, again, can you, as someone who, say, is blood type A, can you receive blood from just anyone you want? Why not? Yeah, exactly. So your immune system uses those glycoproteins to recognize it as either self or not self, and so you can have a reaction where you actually reject that blood coming in if it didn't match up with the glycoproteins that you had coding for your A blood, or whatever it happens to be. And then, again, lipoproteins we mentioned are very important for lipids and transporting lipids around the body. Cholesterol metabolism and transport is going to be one of the main things. You'll see those lipoproteins being used. We'll talk more about those in detail later on. So some other functions that we can have for our proteins, some of them they can be structural in nature, so things like collagen fibers. You'll find these in like connective tissues. The keratin in the skin is made up of proteins that are able to provide kind of structure and support for ourselves. Enzymes, you mentioned, help to assist in lots of different chemical reactions. We'll talk exhaustively about different types of enzymes as we go forward. And then antibodies are another thing that would be really, really important here. So antibodies are able to bind to specific targets and either recognize those as either self or not self, right? And so when an antibody binds to something, it says, hey, I don't recognize this. What does it normally do? Yeah, it brings back up, right? It'll activate the immune system to actually help to target that, right? So that's why you have antibodies against specific disease states. It's how you develop immunities by having these antibodies, and you help to activate the immune system and say, hey, that's varicella, or hey, that's pneumococcus. Let's go ahead and attack this, and that way you don't get sick in the first place. Other things we can use proteins for include receptors. We'll talk about these again ad nauseum when we get to the pharmacology sections. We deal a lot with the receptors. Basically, receptors, they're going to be responding to specific, basically think of a lock and key kind of mechanism where the receptor is going to be the lock, and specific molecules can come by and activate these, right? So you can have insulin receptors, you can have norepinephrine receptors, all different types it can respond to, and then catalyze certain reactions within the cell itself. So for instance, we'll talk a lot about secondary messenger systems, both here and in pharmacodynamics, but by having, say, a molecule, or a ligand as we'll call them, binding into these receptors, it will cause a secondary activation of a process within the cell, and then all sorts of things can happen. So either cause a cell to move or to produce a new protein, whatever happens to be basically kind of a secondary step to have that occur. And then also we'll have different carriers. You can see these either being within the cell membranes. Certain molecules have a hard time crossing cell membranes by themselves. Either they are too hydrophilic and they're not able to cross that lipid bilayer, or they're too large to get across the molecule, the actual membrane itself. Sometimes we'll use carrier molecules to actually help that process out and actually get it into the cell by those means. We'll look at some of those carrier proteins as well. Okay, and then looking at our nucleotides, so these are going to be our nitrogenous bases that fall into two main categories. We're going to find we have our pyrimidines and we're going to have our purines. Basically, just think about the purines having a smaller name go to the bigger molecule. It's kind of counterintuitive, but that's how they name them. So we have for our pyrimidines, we have cytosine, uracil, and thymine. What's the main difference between uracil and thymine? Yeah, which one's only found in RNA? Yeah, uracil is only found in RNA, so that'll be important when we're talking about coding for those different proteins we were talking about previously. And then you have your purines, which will include guanine and adenine, and so we'll find out how these interact with one another when they actually get into the DNA and RNA. And then these can be bound to either two different types of sugars. One can be deoxyribose, where you'll find that in the DNA, obviously, and then you'll have just the ribose. So you can see the difference here. Obviously, deoxyribose is deoxide, is missing one oxygen molecule there. So that's the main difference you're going to find between those two. We'll find that these actually become really, really important when we get into talking about chemotherapy, right? So drugs that help to target cancerous cells. Sometimes we'll actually use drugs that can actually modify these sugars. So if you ever heard of a drug called cytarabine or Ara-C, it actually modifies one of these sugars, so that way when it gets incorporated into the DNA, it actually prevents any further elongation. That's how you can cause a cell to die off. So we'll talk more about these in detail, and you'll see how it comes up in pharm pretty frequently when we're dealing with cancerous type cells. So just as a sneak preview to whet your appetite for that later on. Because I know you guys are really interested in getting into pharm, right? Okay, so we mentioned DNA is that deoxyribonucleic acid. So this is going to be our main, where do you mainly find DNA? Yes, in the nucleus of the cell, absolutely. And we'll see how our bases are going to be binding here. Usually a purine binding to a pyrimidine. And again, there's going to be these hydrogen bonds that are occurring between these two. And notice how they're kind of complementary, they kind of fit together. And then we're going to find that we're going to have this kind of superhelical structure that occurs to help kind of save space. Because otherwise it would be very, very long, it'd be hard to store all that information. Again, cytosine's only going to be binding to guanine, and thymine's only going to be binding to adenine, right? What would happen if you had these base pairs not meeting up appropriately? What do you think? Yeah, possibly mutation could occur. Yeah, what else could happen?
It's not uniform. The width is uniform.
Yeah, you could have some issues. Yeah, so basically, if you were to have that problem, we actually have means of correcting for that. You have things like DNA polymerase, an enzyme that can actually come along and proofread the DNA to make sure everything looks right. And if they did find problems like that, it could actually correct that. In some cases, you may find the damage is too much. If you had enough base pairs that were not meeting up appropriately, it could actually signal cell death. You guys know what that process is called? Yeah, apoptosis. Yeah, so it's like kind of programmed cell death, and that's what we do with a lot of our chemotherapy drugs. We try to cause so much DNA damage to those cancerous cells that there's just too much damage. Let's just get rid of the cell, right? It's just kind of sell it all off and be done with it. That was a non-intentional pun, but. So anyway, the DNA will be twisted into this double helix structure. Again, we have enzymes that are also responsible for helping to unwind it when you're going to be doing things like cell DNA replication, or if you're doing like protein transcription, things like that can be utilized there. So things that will help to unwind the DNA and also cause like some single strand breaks. So you can actually cause the double helix to relax itself, and you can actually cause that reading to occur. So lots of enzymes are involved in that process there. But we know it's the basis for our genetic code. This is where all of our proteins are going to be transcribed from. And again, what do you think is going to make the difference between, like, say, like a skin cell versus like a heart cell? Because we all have the same DNA in all the cells, right? So what's kind of the difference?
Which ones are turned on, which ones are turned off.
Exactly, yeah. So you're going to find there's different things that are going to turn on or off our genes to differentiate them, to make a skin cell not produce the same proteins that a heart cell is going to do, that a brain cell wouldn't do. So you guys will get probably more of that in genetics, if I had to imagine, but we'll talk a little bit about it here as well in just a little bit. Again, here you can see kind of the hydrogen bonding that's occurring between our different base pairs. Again, guanine to cytosine, or C to G. Notice how they are very complementary to one another. They just connect very nicely. Again, hydrogen bonds, we didn't talk about those too much. We were talking about different types of bonds, but is it a very strong, very weak kind of bond? What do you think? Fairly weak, yeah. So again, it's not going to be as strong as like a covalent bond. Again, covalent bonds where they're actually sharing those electrons and they're very much tied to one another. Here you can see that it's a relatively weak bond. There's still that force of attraction, right? Because we mentioned that, especially you know, the H being on the positive side, the O is going to have that more negative conformation there, and so they can kind of attract one another. It's good. We don't want these to be covalent bonds, because what would that mean for us? We could never pull them apart to cause, you know, transcription to occur. So we want to have that kind of a little bit weaker bond in order to be able to let it be more functional, let us to be able to do things with it. Then again, thymine, adenine, again having complementary hydrogen bonding here as well. Again, notice the sugar phosphate backbone is going to be present throughout this. As we will see when you talk about actual production of these different base pairs and whatnot, there'll be another step where some of our chemotherapy drugs can actually fit in and inhibit this. But again, you need to have the sugar phosphate backbone connected to the base pair. If you don't have that, you cannot cause elongation of the DNA, and you'll have kind of early termination that can also lead to apoptosis there. Okay, then on the flip side, we have our RNA, which is going to be our ribonucleic acid, basically just using a different sugar here, and also we're going to see that it has uracil instead of thymine. What do you think uracil is going to bind to? Yeah, so we're going to bind to adenine, so basically just as a stand-in for thymine there. And again, usually going to be single-stranded instead of double-stranded, right? Because this is going to be much more kind of functional molecule. We're going to code for something, send it off to different parts of the cells in order to help produce proteins and whatnot. Again, just looking at the comparisons between two molecules here. Again, deoxyribose and DNA versus ribose. Again, just one more oxygen there. And thymine versus uracil. Very similar molecules, but the differences there are important to note. Okay. Again, uracil only binding to adenine, so that's the main difference there as far as binding goes. But again, still a pyrimidine, still having the same type of hydrogen bonds you can see when it's actually linking up with the DNA to form that the elongation of the RNA. Okay, so that concludes the first section. Any questions I can answer for you guys? All right, pretty basic review. Any new information you guys have gleaned off of this? Both a little bit, maybe? I see no nods. I'm assuming, okay, you guys knew everything about this. Awesome. You guys are going to do great on the test. Next, we're going to continue on with cell structure and function. Also should be a decent amount of review for you guys. Okay, so we're going to talk about some of the different structures within the cell. We'll talk about different functions that can occur here as well. We'll also look at protein synthesis in regards to how RNA is formed and how it goes on to produce proteins. And then we'll also talk about enzymes and energy production. And then the next lecture we'll actually get into more strictly how energy is produced within the cell. So we're going to get into our Krebs cycle and all that fun stuff. So if you guys ever had nightmares about having to know your whole TCA cycle, those are going to come back for you. Become true. No. Okay, so again, our objective here is to just understand the organization and functions of our cells. Okay, so here's a cell. Everyone got that? All right, good. All right, so we know that these are going to be the basic functional units of the body. We're going to find that the primary components of a cell, we have our plasma, cell membrane. Again, that's going to be that lipid bilayer we've been talking about. It helps us separate the external versus the internal environment, right? So it's going to be semi-permeable. Some things can cross over the cell membranes, but a lot of things cannot. And that's important because we don't want just anything getting into our cells. We don't want everything just leaving our cells. Cytoplasm will be the actual fluid compartment within the cell itself. Organelles are just going to be our functional proteins and different things like that are going to be doing different functions, as we'll get into in more detail. And then finally the nucleus that contains the DNA and kind of directs a lot of the cell activities. Does every cell have a nucleus? No, which kind of cells don't have nucleus? Yeah, red blood cells is a really good example of that. Do any cells have multiple nuclei? That's in the question, so probably, right? So what type of cells? Yeah, muscle can actually have multiple nuclei within the same cell. So again, there's going to be some exceptions to this, but in general, mostly one nucleus per cell. Okay, so here are the different structures that we will talk about in more detail as we go forward. So again, I'm not going to just read through this whole thing. We'll talk more about it in detail as we get to the individual slides, but this is a good kind of reference back to make sure you've kind of studied through and are familiar with all the different components here. Hopefully nothing should be too new for you guys since you've all had similar class before. Okay, so our cell structure, we mentioned that with our membrane proteins, these help to provide several functions. Some of them can be just structural support, can kind of help strengthen that lipid bilayer to give a little bit more form to it. And then we'll see that transport is also going to be very, very useful. We'll see that many water-soluble substances cannot cross into the cell. And why do you think that is? Something is water-soluble, just can't cross that lipid bilayer. Why is that? Exactly, yeah. So if something is hydrophilic, if it's very water soluble, it's not going to be very lipophilic at the same time, right? So it's going to have a hard time crossing that lipid bilayer. It may be able to get to these polar heads, right? That phosphate group that's on these phospholipids, but it can't cross this the fatty acid portion, right? So because it can't cross that, water soluble molecules, things like glucose, are not able to freely cross over there, right? So that can be very, very important because we don't want every single bit of glucose into our cells. We don't want every single water soluble molecule to cross over. So how do things like glucose actually get into the cell then, if that's the case? Yeah, so channels, carriers, transporters, however you want to call them, like there's different means in order to facilitate that. So a lot of cases we'll talk about different means to get things into the cell. Some of it can be active processes, some of these will be passive processes, and so we'll look at those in more detail. Other things that we can find on the cell membrane surface is going to be receptors. You can find that hormones can be really responsible for causing a lot of changes by binding to these receptors, other molecules as well, right? So for instance, if I have a cell that's getting hit with, say, nicotine, it may cause one sort of a response versus if I were to hit it with, say, norepinephrine, right? So different effects can occur depending on the type of receptor site that I'm activating. And will the same, so if I have like nicotine is a substance that causes lots of different cellular processes, will it cause the same response in every type of cell? No, right? Because there's going to be different signal pathways. You can find that a specific type of cells may respond one way, a whole other cell may respond differently, right? So say like something like some of the cells within like the reward pathway within the brain, right? We know nicotine is very addictive. That may activate those dopaminergic neurons in one way versus if it were to activate, say, the skeletal muscle. You're going to find different effects there based on the cell type and what type of signals are actually being caused when those receptors are being bound by nicotine. So you can also find that a lot of kind of the identifiers for cells to show that, hey, this is a self cell, not a foreign cell, are found on the cell surface as well, right? So if you have an immune cell that comes along, say like a T cell here, if it were to come by and bind to, say, a self cell, you have receptors here that are able to identify. Once that lock and key mechanism happens there, they identify as, okay, this is a self cell, I'm not going to attack it, right? Versus if you were to inject, say, like a foreign cell, it would activate these kind of foreign responses on the T cells and say, hey, this is a foreign cell, let's go ahead and activate the immune system, let's get some macrophages over here to eat the cell up, whatever it happens to be, right? So that can be really important to identify other types of cells. When do you think this becomes a problem? What type of disease states do you think could we be introducing foreign cells into a person that could cause problems with this? Yeah, transplant's a huge one, right? So if you were to take, say, someone's liver, say your neighbor's liver, and put it into your body, all of a sudden you're going to have different receptor compounds there. You're going to have different things at the surface, different things like glycoproteins and whatnot that are going to identify someone else's cells. And so your immune system can respond to that. And that's why we have to give things like immunosuppressants to tamp down the immune system in order to allow those cells to proliferate and to kind of work normally without being attacked. And that's called graft versus host disease, when you have that the immune system attacking that foreign tissue, right? So that's that same thing we mentioned. If you were to inject someone else's blood that had a different blood type than you, that would be the same kind of process happening. It's recognizing as someone else's cells, and it says, hey, we don't want this, let's go ahead and activate the immune system.
Host versus graft, or graft versus host?
Graft versus host. Well, I guess the nomenclature doesn't really, you usually see graft versus host disease, usually what they call it. But yeah, technically it's the host reacting to the graft that's being implanted, right? So you may see it called either way, but I usually see graft versus host. Yeah, because I think if you look at like notes and things, you'll see like GVH. You know, that's usually what how they abbreviate it. Yep. Okay. Not all of our nomenclature in medicine actually makes sense. It's a little secret. Okay, so some other ways that we can help get stuff into the cell is going to be a process called phagocytosis and pinocytosis. So can anyone describe this process to me, since you guys are physiology experts?
How in detail do you want? How in detail do you want?
Just give a description. If you're trying to explain to like a layperson, let's say. Good. Yep. So it's just eating like a hamburger, right? So, right. Good. What would be like pinocytosis? Yep. So like cell drinking, right? So that's how it's described to me, like an undergrad biology class, right? That's basically the process, right? That's basically what's happening here. To give you a little bit more detail on that, usually what you find is that there's going to be receptor sites within on the cell surface, and once those get activated by a particular substance, that can be the thing that really triggers either the phagocytosis or the pinocytosis. Basically, pinocytosis is better for smaller molecules. The phagocytosis is going to be better for like large things, right? So can you guys think of any examples where this might occur? Yeah, macrophages are a good one, where it identifies one of those other cells or something like that, or a bacteria or something, and it goes and envelops it, eats it up, and then we'll see that there's lysozymes and things like that that can be useful for breaking tissue like that down. What are some other examples? Iron can be another big one. So the way that we actually get iron within our cells when we absorb it from the GI tract actually is through the same similar process. There's no specific or specific like transport proteins that can get iron into the cells. It can happen through this process. So, but again, it's really important for inflammation, host body defenses, and also for apoptosis. We can activate that process as well when you're trying to get these cells that are triggered for cell death. You have the macrophages come along, eat them up, and then kind of break down the products there. Another good picture of this process happening is the cell's kind of extending the pseudopod, giving it the hug of death, and it's going to envelop it and then be able to do whatever it needs to do with it. Okay, then on the other hand, with exocytosis, what is exocytosis? Getting rid of stuff, right? Or just ejecting it from the cell. Notice here that a lot of times there's a term on that previous slide, all the different cell structures, vacuole. Oftentimes we'll call them vesicles as well. There's not really a whole lot of differentiation there. I'll probably call them vesicles more often. But you can notice here where basically the cell will have a vesicle of some sort of product, whether it be a neurotransmitter, it could be a protein, could be lipids, any sort of different things. Usually are going to be here hanging out in the cytoplasm, and they can be ejected out. Sometimes you'll find this constitutive secretion where as soon as the cell produces it, it's able to send it out really without any kind of particular signal, right? It's really kind of an unregulated process. On the other hand, sometimes it can actually be triggered by a specific binding of a receptor. So you notice here in this kind of bottom portion, you have a ligand, which is just something that binds to a receptor, activating this receptor that causes a signal to occur that says, hey, this cell should be releasing its vesicle right now, right? And so it could be a different product completely. But basically, the vesicle will merge with the cell membrane and then release its contents, and then it can either regenerate that or whatever it needs to do, but basically can get back to its baseline state. Okay. So again, this can be really important for especially a lot of drugs. If you're trying to inhibit secretion of certain substances, or if you're trying to promote secretion, affecting these receptors can be very important there. And we call this regulated secretion because of the fact that it has some sort of trigger for it to occur, right? It's not just something that's occurring all the time or without any specific regulator. This is a specific receptor is being activated to cause this to occur. Okay. Some other cell structures you can find, especially on the cell surface, are going to be the cilia. There's going to be tiny hair-like structures. We mentioned these previously, especially like in the lungs. Those are really good for getting debris and bacteria and things like that by having kind of this constant, concerted effort to move things up through the respiratory tract and get rid of things and keep the lungs sterile as they should be. You can also find primary cilia as well. The eye is another good example where you can find those. Those are non-motile, but they are also going to be important for things like photoreception, right? So being able to see. And we notice there's disease states that can affect these cilia. So we mentioned like COPD, like chronic smoking can affect these cilia and actually can damage them. Sometimes you can actually see the cells being replaced by a more like scar-like tissue that is non-functional, does not have these cilia, and then they're not really going to be able to respond and get rid of all this stuff. That's where you can see like more pneumonias in those patients and things like that because this process is not working right. You get just some other pictures of cilia on there, just basically just little furry cells. They're kind of moving things along. Okay. You can also have some cells will have flagellum. Actually, we only have one cell like this, and only probably actually not quite half of you guys. It used to be very small proportion of people here had any kind of flagellums in them, but it's been increasing lately, which is a great thing. So anyways, this is a kind of a single kind of whip-like structure that provides some motility there, right? So sperm are really the only cell that we have that has a flagellum. But where do you see more flagellum more commonly? What non-human cells, I should say. Yeah, bacteria usually have flagellum because they help to kind of swim them around whatever kind of tissue they happen to be in, right? So that's probably more commonly where you're going to be able to see flagellum. But here in the human body, sperm are really the only ones there. Okay, we also have microvilli. What are microvilli? Yeah, little tiny folds, right? So it almost looks like these like little polyps. And what are their function? What do they do for us? Yeah, so surface area is a big thing here. So we increase surface area, and if you imagine, like if you had the, say like the small intestines, where I think most commonly like the microvilli, if you're trying to absorb the nutrients from the food you're intaking, you want to have as much surface area as possible. If the small intestine was just a smooth kind of tube, you'd only have so much surface area, and all that food and stuff in the center really wouldn't get absorbed very well. But by having these microvilli where the food and nutrients can kind of like mash into them, you increase the surface area quite considerably, and that better absorption of different products. So this can be very, very important. They allow for sometimes chemical reactions to occur, but most often you're going to have rapid diffusion of materials across these cell membranes. The intestines, again, I mentioned, are probably the more prominent ones you guys will be encountering, but also the kidney tubules themselves are also very important for this as well. These help to deal with like regulation of water reabsorption or secretion, electrolytes, and things like that. By having the extra surface area, you can be much more functional. Again, just another picture of some microvilli. So you notice here, if this is just kind of a smooth cell membrane, not a whole lot of actual surface area here, but by having all these kind of folds, you're increasing it very, very significantly there, just allowing for better absorption. Okay, next we'll talk about the endoplasmic reticulum, of which we have two varieties. First one is going to be the granular or rough endoplasmic reticulum. What's the main difference between the rough and the smooth endoplasmic reticulum? Ribosome, yeah, absolutely. So the ribosomes are going to be really important for the rough endoplasmic reticulum. This is where a lot of the protein production is going to be happening. We'll talk more about these in detail just a little bit. But yeah, so that's the main thing where a lot of protein production will be occurring within that rough ER. And then the smooth ER, that's going to be more responsible for things like production of certain lipid substances. You can see that a lot of these kind of the part of the membrane here can actually bud off and actually form new vesicles, right? So that can help with transport. Usually things will kind of go from the ER over to the Golgi apparatus we mentioned in just a second, you can kind of get rid of stuff there. You'll also find that in specific cell types, you can find specialized uses for the sarcoplasmic reticulum. Mainly, say for instance, in the endoplasmic reticulum. In the muscle, it's called the sarcoplasmic reticulum, and it's a really big repository for calcium. And we'll see that calcium is very, very important for muscle contractions. And so, by having that storehouse there, it's able to release a large amount of calcium and then reabsorb it again to allow for kind of successive contractions of the muscle. All right, next we have the cytoskeleton. Cytoskeleton is just helping to provide us kind of good form and function to the cell itself. We allow it to have a little bit kind of more rigid kind of structure to itself. It can reorganize itself very easily, so it allows for the cell to either move or kind of reconform its shape kind of as needed. This will also be important for the spindle formation when we talk about mitosis and meiosis, able to kind of sit there and pull the DNA apart when you're trying to cause cell division to occur. Notice here you also have these centrosomes, and these are really responsible for producing all of these microtubules that help with this process. Some pictures of the cytoskeleton here. You can see how it kind of helps keep everything in place. It's not permanently there. It obviously can shift and rearrange itself as needed. All right. Next we have some lysosomes. What do lysosomes do? Yeah, more often digesting things. So they're able to break down products and help with kind of cellular digestion in a lot of cases. Most often, after you have like something like phagocytosis, where food molecules are being brought in to the cell membrane, you'll often have digestive enzymes in these lysosomes combining with the food vacuole, and then you can have digestion can occur. This can also occur with, like, if you were to trigger apoptosis or something like that. These lysosomes can also be really responsible for that. What do you imagine the pH of the lysosome is? Yeah, so we're going to be a lot more acidic in order to help break down a lot of those tissues or a lot of those cellular components. So it's important that it's kind of sequestered off, because otherwise it would not be able to kind of maintain that nice acidic environment in order to cause that digestion to occur there. Obviously, the primary lysosome is going to be really only containing the digestive enzymes, and a lot more acidic than the cytoplasm. The secondary lysosome is one that combines with the food vacuole, and you have kind of partially digested products there. And then you'll have a residual body where it's kind of undigestible material is there, and then it'll be just kind of expelled out in the cell through exocytosis. Just another example of this occurring. You can see here in this particular picture. I lost my. In this particular picture, you notice here how you have like some cellular components that are old and they want to get rid of them. You can end up kind of budding that off within a vesicle and then having the lysosome break that down and then being able to excrete it. Sometimes you can reuse some of those components in order to help produce new components. You can see that occur sometimes. And again, most often the residual bodies will just be excreted through exocytosis. Okay, you also can have some peroxisomes. These are going to be a little bit more specialized kind of lysosomes, and you'll find these are useful for certain oxidative reactions. And also, a peroxisome is probably going to contain what product, do you think? Peroxide, yeah. So it actually contains hydrogen peroxide. And so what's the chemical structure for hydrogen peroxide? H2O2. Is that a very stable molecule? Not a very stable molecule, but it's very, very reactive, and so it can actually cause breakdown of a lot of different types of tissues. It can be damaging if you have too much of it within particular cells, but the nice thing is it forms these reactive oxygen species, where basically the oxygen is kind of by itself and is very, very reactive, and go and break down things like proteins. Here you can see it's useful in the liver to help break down alcohol through a certain pathway. You kind of see that here. On this point, we have alcohol entering into the liver. H2O2, or peroxide, can be used as this catalase enzyme, and it ends up producing water in that reactive oxygen, and that can help to break it down into acetaldehyde, right? So you guys know what acetaldehyde does in the body? We'll talk about alcohol metabolism, but basically that's the thing that causes, especially if you have a deficiency of an enzyme to get rid of this, you can have a lot of flushing that can occur there. You get very, very sick and nauseous, and a lot of heat and things like that can occur. So not necessarily related to the hangover, but if you ever feel really, really bad and hot and flushed after drinking alcohol, usually do the acetaldehyde. And so peroxide and these peroxisomes are useful for helping to break down a portion of that. I like to talk about alcohol as a good example for stuff, so it'll come up pretty frequently. I don't know if that says more about me than anything else, but it'll come up. Yeah, but then basically it ends up forming H2O. And again, just to show you how reactive hydrogen peroxide is, who has a bottle of hydrogen peroxide at home? Or maybe at their parents' house or something like that. How old is it? We bought it back in the 80s, right? If you pour it out, what do you think's in that bottle now? Just water, yeah. So because it is so reactive, if it ends up reacting with oxygen, it breaks down over time. So that's why it's always important to have a fresh bottle of hydrogen peroxide, because most of the time it's going to end up being broken down over the years, and so you're just pouring water onto your wound or whatever it happens to be. So just keep that in mind. We also had bad cases, just to kind of give you a clinical example of hydrogen peroxide from a toxicology standpoint can be really, really bad, because one of the other things that once that oxygen is freed from the hydrogen peroxide, you end up with water, sure, but you also can end up with a large amount of O2 being produced. So that's actually one of the things that you can give to animals if they ingest something they shouldn't. Give them hydrogen peroxide, it makes a lot of oxygen in the stomach and it causes them to throw up. But you can also see, like, we had one case where we had a young girl, probably like 5 or 6 years old, ended up going and getting into this hydrogen peroxide product from a health food store, and I think it was, normal hydrogen peroxide is like 3%, right? Usually not too concentrated. This stuff was like 70% hydrogen peroxide, very, very concentrated. She ended up getting a large amount of it, right? Because kids get into stuff and end up drinking some, and ended up forming air emboli. Is actually able to form emboli within the blood itself, and so we actually had to put her into a hyperbaric chamber, which is increasing the pressure on the oxygen in there to help kind of shrink down those bubbles, and she cleared eventually. She did fine, but just note that hydrogen peroxide is certainly nothing to mess around with, and is very useful for metabolizing products within, especially like the liver.
So I had a friend who said that they couldn't break down alcohol because they were lacking an enzyme. Would this be what they were lacking?
They probably lack the enzyme acetaldehyde. So I'll talk about more of this probably elsewhere in class. But basically, alcohol, one of the primary ways it gets metabolized is through alcohol dehydrogenase, which produces the acetaldehyde, and then there's acetaldehyde dehydrogenase that breaks that down into, eventually goes into carbon dioxide and water, right? Usually, especially patients of Asian descent, they can have a deficiency of that acetaldehyde dehydrogenase, and that's what they get into a problem, is because they end up building that product up and they get very flush, and they get potentially hypotensive, and they feel really bad, and they throw up, and yeah, it's no good. My medical director in during my fellowship, his name was Dr. Kunisaki, and so he had an Asian background, and he could not drink alcohol whatsoever because he would just immediately get flushed and start sweating profusely, and it's just no good for him. So he just knew, I'm not going to be a drinker. Yep. So there's actually some drugs that can cause this as well. So we'll talk about those reactions later on. But that's probably what the problem was with your friend. Not a problem. It's just that's how they are. Yep. Okay. Moving on. Next we have the mitochondria. We mentioned this is powerhouse of the cell, absolutely. So this is where we have a process called oxidative phosphorylation that can occur here. We'll talk about this ad nauseum in a few lectures when we talk about energy production, but what is the main thing produced here? ATP, which is?
Energy.
Adenosine triphosphate, right. And so why is that energy? You can break it down. Yeah, so the phosphate groups end up having it's a high energy bond between those phosphate groups, and we can use that for lots of different processes for energy. But basically, this is an oxidative process. We need oxygen for this to occur. So this is very good for aerobic metabolism, as we'll also call it. You can actually see problems like certain toxins can actually affect this, especially like cyanide. So cyanide can actually stop this process from occurring. You can't actually utilize oxygen within the mitochondria. So we'll talk about that later. But essentially, we need to have oxidative phosphorylation occur here in order to produce ATP. This is the most efficient way to produce energy within the body that we have. Otherwise, we have to rely on anaerobic processes, which by calling anaerobic, what do you think that means? It doesn't use oxygen, right? And so it's going to be much less efficient. Also produce a lot of acids and heat and things like that. So this is the best process we have. We try to utilize this whenever we can. All right. Next we have the cell nucleus. And again, most cells are going to have a nucleus. We mentioned the RBCs, the red blood cells, are not going to have one. Certain cells are going to have multiple nucleuses that'll be found within them, or nuclei. Again, normally you're going to end up having a nucleolus is kind of filled in the center here, and then you're also going to have a lot of chromatin, right? So this is basically just kind of this unwound kind of DNA material. It has not been really kind of super condensed into the actual chromosomes like we're used to seeing when you see like cell division and things like that. So it's kind of unwound, more free to be utilized by RNA and things like that in order to help produce new proteins and whatnot. Again, the nucleolus itself does not really have a membrane around it kind of separating itself, but it's where you see a lot of RNA and a lot of the proteins that are going to eventually be in ribosomes are found there. And you can actually see this start to swell whenever you're about to have cell division, right? Because you're at a point where you need to produce a lot of new proteins, a lot of new DNA, so that can actually end up swelling during those processes there. And so we'll see this a lot more when we talk about things like mitosis and meiosis. All right, Any questions from the first half? I should say the first quarter, since it's going to be a long day for all of us. I'm just kidding. No, it'll be fun. All right, so moving on with our ribosomes and talk of protein production. We know these are the protein factories of the cell. We mentioned they're going to be more on the granular endoplasmic reticulum there. And really, this messenger RNA is going to be the primary shuttler of the genetic information from DNA over into the ribosomes. And so, for instance, here we have like the 70S ribosome. It's useful for producing new proteins. One of the interesting things you'll find when we, or maybe I find it interesting, is when you get to pharmacology and we're talking about antibiotics. A lot of antibiotics actually work on inhibiting ribosomes. Right, actually prevent protein production in a lot of bacteria. And so you ask, well, how come the antibiotics work on bacteria, but they don't necessarily affect us quite so much? Do you guys know why? Yeah, we actually have different ribosomes. So because we have different ribosomes, we have antibiotics that can be more selective against bacteria versus affecting us, right? Certainly still cause some side effects. But that's one of the big things you'll find, is, especially with medications, like by having selectivity for specific targets, you can help to have clinical effects, which is killing off the bacteria, versus having negative effects on us, hopefully, which we want to produce proteins. That's a good thing. And so that's one way we can kind of differentiate between self and bacteria. So the generic kind of process that's happening here is you have the nucleus of the cell. DNA is going to be read by the RNA over through the DNA transcription over to have our RNA. You'll have different splicing that occurs. We'll talk about introns and extrons and all that good stuff. But eventually you get this final mRNA product here, and that's going to be shuttled outside of the nucleus and be transported over to the ribosomes, where it can be interacted with like tRNA and all of that to produce new proteins. Basically, they'll just end up reading 3 codons at a time. That's going to code for a specific amino acid, and then that'll just be added on to the growing protein chain. Right? Protein chain can then be either utilized for whatever process, or it can be cleaved and changed and whatever to get a final kind of functional protein there. That's kind of the kind of generic process that happens there. So how do we control protein synthesis? We've alluded to this previously during this lecture. But essentially, we mentioned that there's over about 30,000 different genes contained in each cell, and how we differentiate between, say, a skin cell and a neuron are all going to be going back to what's being promoted, what genes are turned on, which ones are turned off, and that's going to be affecting the type of proteins that are being produced there, right? So that way we can differentiate between these different cells. And so the RNA molecules are going to be really useful for helping to encode for those different proteins there. Notice here you have our DNA strand. Normally you're going to have different enzymes come along. They can basically unwind the DNA, cause a single strand break. We'll talk about the different enzymes later on, but basically you're going to be able to have a single strand, read that, and then based on the different the pairing there between the RNA and the DNA, you'll have a single strand of RNA that's being made there, and basically you'll have your transcription happening there to eventually produce your new protein. So you can see how based on reading the DNA strand, you're adding on new base pairs to the RNA molecule, right? So it's going to be continuing to extend. Eventually it'll end up hitting kind of a terminator sequence where it's like, okay, this is long enough, we're done. Let's go ahead and send this off for eventual translation. So again, these are going to be based on these triplets, or these codons that are going to be coding for these amino acids. So for instance, if you have a G, guanine, adenine, you're going to end up getting glutamic acid. If you have uracil, cytosine, uracil, you're going to end up getting a serine. Obviously, every codon, multiple may be coding for the same amino acid. Some of them may be coding for no amino acids whatsoever. Some of them may be doing other processes there. So based on the sequence, we can either get our protein being made, we may getting like a starter sequence, a stop sequence, just depending on where it's reading it at within the DNA molecule itself. So again, that transcription is going to be that process of going from the DNA to producing your RNA. The enzyme that's primarily responsible for this is going to be our RNA polymerase, right? Normally, DNA polymerase is responsible for producing new DNA. RNA polymerase is useful for producing this new RNA. It's going to bind to those promoters. Usually, promoters are going to be ahead of the gene to be transcribed. And again, I'm kind of simplifying this down to our purposes. I'm sure you guys have gone into much more detail on this in previous classes, but it's not really germane to our discussion. And basically, by reading these promoters, RNA polymerase is going to say, okay, let's start producing a new RNA strand. It's going to start reading these base pairs after the DNA helix unwinds and then separates, and then basically it'll be finished once they hit that chain terminating sequence. It's going to read a certain sequence, say, hey, we're done, send off the RNA to be processed. Bless you. So first off, we have this precursor messenger RNA, this pre-mRNA. Notice here that the RNA structure that is produced after reading that DNA, whatever gene it happened to be, has a lot of excess information we don't necessarily need, right? So by cutting out the introns, these are going to be removed by splicing the RNA pairs together. You're left with these exons here, right? So you have your initial strand here, finish it up on the end. You only have kind of the necessary bits left here, and that's when you get your messenger RNA essentially. You can also have small nuclear RNA. These are going to be little bits of RNA that can help to deal with the splicing, right? They can recognize those structures that need to be cut out and kind of direct that process there. Once you have your mRNA, that's going to carry that genetic code over to the cytoplasm, and it's going to be helping to decide what type of proteins are going to be made there through that translation process. Those here are going to be kind of entering into the ribosomes based on the codons. The transfer RNA is going to be brought in here, and we'll add on, continue to add on to that strand of protein that's being made there based on the 3 codon sequences reading. It'll produce a specific amino acid to kind of come in there. The tRNA has to interact with the specific codon for the RNA, and that produces dictates what protein's added on, or which amino acid. You can also have some ribosomal RNA. It's going to be mixed in with the ribosomes that kind of help direct processes there, and you also have some microRNA as well. They don't actually code for proteins themselves, but they can actually help to deal with gene transcription and regulation, deciding which genes should actually be made into proteins, which ones shouldn't. So you notice here you can have multiple proteins that are being made from the same strand of messenger RNA as it gets fed through multiple ribosomes, right? So there's one long strand. As it goes through, the tRNA comes in and is going to add on those amino acids there. Okay, so any questions on that? So again, pretty basic, but it's kind of useful for our circumstances here. Next, we'll talk about our cell cycles. And so the 2 primary things we're going to be talking about are going to be mitosis and meiosis. How would you describe the difference between the 2? Yes, okay, so meiosis is for sex cells, right? So it'd be, what's the difference between like the sex cells versus non? Yeah, they're going to have half the genetic information as something that went through mitosis, right? So one of the things I try to do for you guys is like, how would you describe this to someone who was like a layperson, right? Because patient education is going to be a huge thing of what you guys do as a professional, and so if you can't communicate some of this information to your patients, it's not going to make it over to them, and that can really have profound effects on their care. So this is why I try to ask those kind of questions. It may seem silly, but how would you describe this to a layperson? Like, always think that to yourself. If you can describe it to someone who has no medical knowledge, you probably know the information pretty well, right? So that's why I try to ask those kind of questions pretty frequently. So the cell cycle is going to be basically divided into a couple different phases here. So we have our interphase, which is going to be composed of several kind of subphases. We'll have the G1, the S phase, and G2 phase. We'll talk about those in detail. You have the actual mitosis phase, and then the cytokinesis. You guys know what cytokinesis is? Yeah, so the actual the cell kind of buds off, and now you have 2 identical cells, at least during the mitosis process. So during the interphase, you're going to find that lots of RNA is being produced, lots of proteins are being produced from that RNA, and this is where kind of a lot of that synthesis is happening here. Now, a lot of cells who are not actively dividing go into the G0 phase. And so what do you think is happening during G0? Yeah, nothing's building up. They're just kind of doing their work, right? So if it's a beta cell in the pancreas, it's just producing insulin, just kind of doing its thing. There's no actual cell division that's happening there. So you can find some cells that very rarely go into the G0 phase. So you have some rapidly dividing cells. Think about things like your hair. Think about your nails. Think about the GI epithelia. You're constantly kind of sloughing off those type of cells. So they constantly need to be in a state of reproducing, right? So you have some cells that are going to be constantly in that state of mitosis. But other things like neurons, like you don't produce a whole lot of new neurons, especially once you're an adult. So that's going to be primarily in the G0 phase, just kind of doing its normal function.
Would that be like the resting phase that you do?
Yeah, so we call that quiescent phase, where basically it's just resting, it's just doing its thing, right? It's just not in a state of actively dividing. And so this is actually really important because when we are looking at, say like we're talking about cancer, right? So what is cancer? Kind of a baseline description. Yeah, uncontrolled cell growth, right? So if you imagine those cancerous cells, which phase are they usually in? Are they in G0 or not? No, they're never in G0, right? Because they're constantly being dividing because of whatever process has gone awry, whatever mutations happen. They just say, hey, we need to constantly divide. And so when you actually look at when we are giving things like chemotherapy to kill off of those cells, we're also killing off other rapidly dividing cells that aren't in G0. So that's why when you're looking at the side effects from a lot of cancer drugs, you find that things like nausea, vomiting are really important, or diarrhea are really important because of the fact you're hitting those GI cells that are rapidly dividing. What happens to the hair of someone who's receiving chemotherapy? Falls out, right? Because you're killing off all of those rapidly dividing cells there as well. So again, we like to have some selectivity for those cancerous cells, but oftentimes we cannot do that because, again, there are cells. It's hard to really target those. So just keep that in mind, how cells being in G0 can sometimes be protective against specific things like chemotherapy drugs and whatnot. So again, once they're in the G1 phase, this is going to be the growth phase. You're finding a lot of the organelles are starting to be copied in order to get prepared for that cell division that's actually happening there. And actually what you'll end up finding when we talk about chemotherapy is that certain drugs will actually be affecting the cell in different phases. You'll find ones that work specifically in the S phase, some work in the G2 phase, and we can actually target certain cancers by targeting those type of phases. Once the organelles are copied, then we're going to go into the S phase. This is where we have DNA replication, or that doubling of all of our chromosomes. This is usually done by DNA polymerase, right? DNA polymerase does lots of things, not only producing new DNA, also does a lot of proofreading to make sure that the DNA that's copied is correct, right? Because if it's not correct, what happens? Mutation, yeah, so absolutely. So if you have a mutation there, that could lead to cancer, or it could lead to cell death, or lots of different things that could not good for the cell. So DNA polymerase is also useful for doing that kind of proofreading process. And then once we hit the G2 phase, the chromosomes start to condense, right? All that chromatin that's in the nucleus starts to kind of get condensed down, and then we're going to have our actual chromosomes form there. The two strands get plopped together, right, into matching pairs, and so those are going to be done by the centromeres, right? So they're kind of bound there by the centromeres holding the two copies together. Okay. So this is why you get to see, when you see rapidly dividing cells, normally you end up seeing the DNA or the chromosomes in that kind of formation. Okay, next when we get into actual mitosis, we'll see it's going to be divided up into 4 phases: the prophase, the metaphase, anaphase, and telophase. So I kind of call it PMAT. That's how I remember the sequence there. So again, once we had growth of all the organelles, produced the new DNA, trying to get the final growth there and getting all the chromosomes ready to go, this is where we start to enter the mitosis phase. So talk about more of those in detail. Okay, so again, when we're having mitosis, we're having that cell division. Again, the two copies are going to be diploid cells or diploid cells. Those are going to have the exact same amount of chromosomes as the original parent cell had, right? So we're the main process there. We're doubling the DNA and then evenly dividing it between the two new cells there. So again, is there any kind of new genetic information being exchanged, or any kind of change that should be happening there? Shouldn't be, right? It should be identical cells. Should be just clones of one another. Are there changes that can happen?
Yes.
Absolutely, right? So especially in those rapidly dividing cells, sometimes mutations can happen. If you think about things like, DNA damage that can happen from the sun or from other types of radiation, things like that, mutations can occur. But hopefully, in a normal process, we're just producing two new clones of the same cell. Okay, so the first we have the prophase. This is where we're going to be, the chromosomes again are becoming more visible as they get kind of super condensed and down to their chromosomes are being held together by the centromeres. And then that nuclear membrane in the nucleolus starts to disappear, right? So that way they kind of have kind of more free reign to kind of move within the cell itself. The centrioles, right, we said those are important for producing those the tubules in order to help kind of pull those apart, and the spindle fibers start to form from those centromeres, right? Centrioles, sorry. Next, we're going to have the metaphase. Here, the chromosomes are going to start to line up within the center of the cell, and when then they've been attached by those spindle fibers as well. So you can kind of see that. Right here, notice they're being bound up. They're kind of organizing themselves nice and neat in order to get ready for that division process to occur. You can see some actual cells here where that's happening as well, and kind of getting ready for that process. Next, we'll have our anaphase. This is where the centromeres start to split and the spindle fibers shorten, and that's going to start pulling those chromatids apart, right, to the opposing sides. So that way, hopefully, you're going to get one set of the chromosomes on one side, one set of chromosomes on the other side there. And then finally, with the telophase, that cytoplasm is starting to be divided. That's where that cytokinesis actually occurs. And then now we have two brand new cells. The nucleus and the nucleolus start to kind of form again to kind of partition off the DNA, and then chromosomes are going to start to lengthen out in that chromatid into the nucleus. So pretty straightforward process should be happening here through the 4 phases. So what can happen as far as cell death goes? There's a few things that can happen here. One, how would you describe like necrosis?
It doesn't get enough oxygen in the blood.
Yeah, yeah. So that can be certainly one thing that can cause necrosis. So usually hypoxia or a lack of oxygen getting to the cell can be one of those things that causes necrosis. And then we talked about apoptosis as well. And what do you think is kind of the difference between those two processes there?
It's a little bit more clean than the others.
Exactly. Yeah. So which one's more clean? Yeah, the apoptosis is going to be more clean because it's triggered or signals cell death, right? That's saying, hey, we've got a problem here. We need to go ahead and kill the cell off. You can do it in a much more organized fashion. We can end up having, you know, the phagocytes and macrophages and things like that coming aboard to try to bind up these different, you know, residuals of the cell and bind, you know, break them down very easily. It's a nice and ordered process. When you have necrosis like this, when you have you know, say a lack of oxygen getting to the tissue itself, it kind of just breaks open and just kind of releases all its products here, right? And so what do you think could happen if that occurs? Possible risk for infection, possibly.
Spread it. Spread the lysosome contents and everything else that was in the cell.
Yeah, so basically all that stuff just gets lysed out and just kind of gets released into the tissue, right, and the surrounding bits. That can be a good way to cause inflammation as well, right? And so also if you think about, like, for instance, one of the things that we run into with our chemotherapy patients, I'm sorry to keep coming back to the cancer reference, but this is pretty applicable here. When you have a case where you're giving chemotherapy and you're killing off all these cells, you're not necessarily triggering apoptosis in a lot of cases. Some of them are just kind of lysing the cells open. A lot of the uric acid that's utilized in the DNA actually gets released into the bloodstream, right? And so we call this hyperuricemic syndrome that can occur and cause the kidneys to shut down, cause all kinds of problems. And so it's occurring because we have all these cells that are just kind of lysing all at one time, just spilling all their contents into the blood. So that can be a really big problem we can see with those type patients. So again, we like apoptosis if we can have that occur, but in a lot of cases you may end up with this necrosis factor that can be much more kind of pro-inflammatory, just not as easy on the body as apoptosis would be. Okay. Another feature here, the difference between hypertrophy and hyperplasia. Who can describe the difference between those two?
Hyperplasia is increase in number, hypertrophy is increase in size.
Yeah, I mean, it's hyperplasia and hypertrophy. I don't know if I might say that wrong.
Increase in size.
Yeah, hypertrophy is increase in size. Hyperplasia, the cells, yeah. So big difference between that. So if you think about like a hypertrophic heart, you know, someone who has like congestive heart failure or like chronic high blood pressure, you end up seeing the cells that are trying to grow in response to that. Right? The muscles are probably one of the biggest things that do hyperplasia. I'm sorry, hypertrophy. They're going to be increasing in size. You don't necessarily have new cells, but certainly have a lot bigger cells to do whatever the function of the tissue is. On the other hand, you have hyperplasia. That's going to be increasing the number of the cells. Again, that's where we're going to see the most growth for a lot of the body tissue. So think about, like, you know, an infant or something like they're having a lot of hyperplasia of that tissue in order to produce new cells and things like that. Okay, moving on. Next we have meiosis, and so meiosis is going to produce what for us? Yeah, so the big difference here, instead of just ending up with 2 cells, we're going to end up with 4 cells, and we call them, they're not diploid, they're haploid, which means what? Yeah, they only have half the genetic information needed to produce a totally functioning cell. So we'll talk about the process here. Notice it's going to be kind of a 2-step process for the cell division, right, that occurs here. And what's kind of the nice thing, or kind of one of the benefits of going through this meiosis process, like, you know, what kind of happens to this genetic information that gets produced in these new kind of sex cells? Yeah, there's going to be some crossover there. This is where we can have new mutations that can occur, which can be beneficial, right? Because again, if we were just producing the same offspring over and over and over again, that wouldn't be very good, right? We want to have some kind of genetic variation in the offspring that we're producing. Otherwise, we'd be living like, you know, in backwoods, you know, Arkansas or somewhere, and just, you know, the reproductive pool is a little shallow there, and so they have some process. There's actually a case, I believe it was in Arkansas. Basically, there was a family. You can actually look this up. You can look up the Blue family, and essentially what had happened was that they were kind of this family was in the middle of the woods. They didn't have a whole lot of people that they you know, could court, and so unfortunately they ended up kind of reproducing within their own kind of genetic pool pretty frequently throughout the generations. And so what actually ended up happening was there was a mutation where they were lacking an enzyme called methemoglobin reductase, and basically they ended up developing this process where they developed methemoglobinemia. And so if you ever see a patient who has that, they basically look like a Smurf, right? They're still breathing, but they have blue skin essentially. And so because of that, they didn't have enough genetic diversity. They were able to pass on that kind of bad copy of the gene onto their offspring. So if you look up pictures of it, it's kind of interesting because they're just a bunch of blue people. They're not cyanotic, but it's just they have a high concentration of this methemoglobin in their bloodstream. So a little interesting point there. I come from Interlochen, Florida, where you probably have similar processes that are happening there. So just also meth labs, but it's neither here nor there. Anywho, so again, notice whether the difference between males and females. Right. So you notice this little tiny guy, this little kind of puny chromosome here. You guys are stuck with the Y, right? So we get this little tiny guy versus the females are going to get 2 copies of the X chromosome. This is going to have big ramifications for different types of disease states, especially ones where males are only inheriting the X chromosome from who? The mom, yeah. So you can see moms will be carrying some genetic conditions that that will be recessive conditions that normally in a female get covered up by a more dominant gene, but they can be passed down to the male because they don't have an extra X copy to really kind of override that. So we'll talk about things like color blindness. Oftentimes that is being acquired from the mom, things like that. Again, one time I was on the internet and I saw someone has a comeback, says like, "Oh yeah, how many chromosomes do you have? I bet I have more than you." It's not a good thing. You just want this many, right? This is all you want. And how many chromosomes do we normally have? 46 in total, right? So we have 2 pairs of everything, so 23 pairs. Again, one of those pairs being our sex hormones, so either sex chromosomes, either being X and Y for males, X and X for females. Okay, so during this meiosis process, we notice again we have this homologous chromosomes, again 23 pairs, again one of those being the sex chromosomes, X and Y or X and X. In this meiosis process, they're going to be producing the gametes, either ova or sperm, depending on your orientation. We're talking about meiosis 1 and then meiosis 2 to divide these processes up so you can see some of the differences there. Meiosis 1, you're having this reduction division. Basically, the prophase is going to be very, very similar to normal mitosis. The big difference here is you're going to have this crossing over phenomenon where you notice here that the DNA, as it gets lined up with each other, they're going to be have some crossover there. This is where you get genetic diversity, right? So we're able to have some changes there, some mutations that can happen, but basically kind of just mix things up a little bit so that we're just not producing just clone cells. And then again, the process again is going to be very, very similar across them. Once the crossing over happens, then they're going to be able to be pulled apart and go through the normal kind of PMAT process there during the initial cell division. In anaphase, the chromosomes will start to be pulled apart by those spindle fibers. The telophase, they are going to be separated out, and then eventually you're going to have that cytokinesis and have basically 2 daughter cells that are being produced there, right? So again, these are both going to have a full complement of genetic information and have the same 46 chromosomes. They might just be varied up a little bit in regards to the organization for some of those genes. Okay. And then in meiosis 2, this is where we're going to have this process where we're not having another replication of the DNA. All the DNA that we need is already there, but you're going to find that the sister chromatids are going to line up in the center, they get pulled apart, and then we have 4 haploid cells, right? So 4 cells only have half the genetic information we need. For guys, they're going to turn into sperm, and for females, they're going to turn into the ova. Again, just another picture kind of showing the process happening here. Again, this crossover is going to be happening during that first initial kind of metaphase, and then we're going to have this division during meiosis 1, and then during meiosis 2, again, just going to be pulling apart, just no additional reproduction of that DNA between there. Okay, pretty straightforward. Again, just showing an example of how the chromosomes are going to be kind of divided between these two, right? So again, when you're looking at mitosis, again, these are going to be same exact chromosomes, no changes have occurred here, versus here we're going to have some variation, right? So we're going to have the crossing over process happen. Okay, so any questions on cell division? Okay, hopefully the cancer stuff kind of provides some kind of context for how we utilize some of this information. It helps out kind of in the clinical setting a little bit. Okay, next we'll talk about enzymes. So enzymes, what do they do for us? Speed up a process, right? What kind of process do they do? all kinds of stuff, right? You're absolutely right. So everything possible can be helped out with an enzyme in a lot of cases. So it's really important to realize that enzymes are specific for certain substrates, and they are very specific in their function, right? So say, for instance, you guys are familiar with the neurotransmitter acetylcholine, right? Everyone's heard of that probably. There's an acetylcholinesterase, which is an enzyme that only works to break down acetylcholine, right? You get acetyl-CoA and choline basically, and you can recycle that. But acetylcholinesterase only affects that particular product, right? And so it's, again, a very kind of lock and key kind of mechanism where it's looking for a specific substrate, catalyze that process, and then it can kind of go on and do its process again, right? Sometimes you'll find that when the substrate binds, it'll be a cleavage process. So I mentioned that with the acetylcholinesterase example, where again you'd have acetyl-CoA and choline that are being formed from that. So that's one process that can occur via enzymes. You can also have some cases where you have new products being formed. So you have two different substrates that will actually come together, and the enzyme will catalyze a reaction where you actually can put those two. So imagine like RNA polymerase, right? They're going to be adding on new codons there, adding new base pairs when it's reading the DNA. So that would be another case where an enzyme is causing new products to be formed rather than breaking them down. Again, how do we normally know if a product or if something is an enzyme or not? How do you know just by looking at the name? Yeah, so usually has an ASE at the end of it. So we talked about lipases, talked about proteases. ACE on the end of that is always going to be typically an enzyme that we're dealing with. So again, they increase the rate of a reaction, right? And they usually don't change the actual nature of it itself, right? The reaction probably could have occurred by itself, but it may have required a lot more energy or a lot more time in order for it to occur. So this just basically helps to facilitate things going on. And again, the enzyme in a lot of cases are going to be used again, right? So you can find that enzymes can be used to catalyze many thousands of reactions and eventually gets recycled. But again, they're going to be able to catalyze many, many different reactions. It's not just a one and done kind of thing, right? So oftentimes you're going to find that there's an activation energy or there's a specific kind of set point where enzymes kind of work best. And so again, this is why we look at things like the pH of the body. We look at the temperature of the body, because once you start to get outside of the norms, either go too hot, too cold, too acidic, too basic, things like that, you can tend to find that enzymes don't tend to work quite as well, right? They kind of have a sweet spot. They're going to be working the best. This activation energy is where the reactants tend to work best here in order to cause the reaction. And again, if you think about things like fever, right? So we're increasing the heat of the body. Is that a beneficial thing? Actually, fever is probably good, right? It actually helps to catalyze a lot of these reactions, help the immune system work a little bit better. A lot of people have fever phobia, they say, where working in a peds ER, I see this all the time where moms will bring the kid in, they've had a fever for 6 hours. You got to help them. And they're like, they're fine. It's going to be fine, right? So it's oftentimes what you have to do to kind of help out some of these patients. Even my wife, like, she's a pharmacist as well, and when my daughter developed a fever for, say, like 2 days, she's like, I think we should take her in. I'm like, it's okay. She's probably got a virus. Fever's good for her. She's going to be able to work through it. She may be a little bit miserable. May give her some medications for it, but she's going to be fine, right? And so this is a process you run into where a lot of people are just very, very worried about fevers, but they can be useful, especially in the case of fighting off infections due to virus or bacteria or whatever it happen to be. But just know that enzymes tend to work specifically, they work best at a specific temperature, specific, even things like osmolarity, things like, pH can really affect how well an enzyme is going to be working. So it's important to make sure we're staying within that kind of sweet spot. Again, these substrates are going to be looking for specific, or the enzymes are going to be looking for specific substrates in order to make them work. And just think about a lock and key kind of mechanism here. So say, for instance, you have like the enzyme lactase. What do you think lactase does? Breaks down lactose. Absolutely, yeah. So if it's looking, lactase enzyme, you see here, when it sees lactose, it says, hey, this is, it's going to fit my receptor pretty well. It's going to bind to it, and then it can break it down into glucose and galactose, right? On the other hand, if you had sucrose, which would be basically two glucose molecules stuck together, it's not going to be able to fit into that receptor very well, and thus it's not going to catalyze that reaction. Okay, so usually enzymes are very specific for a substrate that it's looking for, right? Obviously, if you don't have a lot of lactase, what happens? Yeah, you're lactose intolerant, right? Or as my cousin likes to do, eat a bunch of cheese and pizza and ice cream, and then it makes us suffer for the weekend. Don't do that. It's just me. Anywho, so again, that substrate is going to bind to the active site of the enzyme. When you find that enzyme, if it shifts or if it gets denatured or changed at all, it can lead to lesser activity, since it can't really bind. The receptor site is going to change to some degree. Here's an example of the enzyme kind of weakening original bonds between a substrate. So say, for instance, the process can either go either way, where you can have, say, substrate A, substrate B coming together, and then you end up having, say, product D now being formed and product C here as well. So you can cause all sorts of different changes with the substrates that are binding to that enzyme. Again, is this usually a pretty quick process or a pretty slow process? Pretty fast, yeah. So a lot of these enzymes are trying to work very quickly. They're helping to make these processes just happen more quickly than they would on their own. Okay, again, we mentioned that enzymes typically have the suffix ase or ase on the end of those. And so, for instance, if you're looking like at a phosphatase, they can work by removing phosphate groups. That makes sense. Dehydrogenase, yeah, it does. Moves hydrogen from those atoms, right? Hydrolases can add water. Kinases, this would be really important for cell signaling. That can be adding phosphate groups to things. And even like isomerases, which help to kind of rearrange molecules into kind of different conformations. So all kinds of different enzymes. You guys think of any other enzymes? Off the top of your head? Lipase, yeah, I mentioned that. Amylase, yeah. DNA polymerase. Yeah, heliase. Okay. Yeah, so all kinds of different enzymes. You could probably could have stringed together any number of syllables together, and I probably would have been like, yeah, that sounds like an enzyme. It's probably out there, right? So that's the thing. So there's lots of different enzymes that are available. But again, usually by looking at the name, you can get a sense for what the enzyme itself is going to be doing. And we'll look at a number of those as we go through the more specific kind of body functions. Okay, mentioned this is going to be influenced by temperature, pH. One of the things we actually run into in a lot of cases, especially if you look at a patient who has sepsis, like what is sepsis? Usually due to an infection, basically kind of whole body inflammation, the whole body's kind of shutting down in more severe sepsis. But in those cases, the patients, do you think they become more acidic or more basic? Typically, they become more acidic because you think about like a septic patient, their blood pressure gets lower, they're not able to shuttle oxygen around to the body appropriately. They end up getting very acidic in a lot of cases, and that can actually affect the process for these enzymes to work, actually make them work less effectively. Some of it may be due to lack of energy to hit the activation energy to cause the reaction to occur. Some of it may be due to actual conformation of the protein changing, right? And so that may lead it to not bind to a substrate as well and to work less effectively. But just know that usually for any enzyme, there's a sweet spot, and if we can stay in that range, it will be working kind of optimal efficiency. Also, the concentration of the enzyme and the substrate is really important. So say, for instance, if you don't have any substrate around, what's the enzyme going to do? It's got nothing to bind to. It's not going to do anything. And then on the other hand, if you don't have any enzyme around, substrate, you could have a ton of it, but it's not really going to bind to anything either. This is actually a problem we run into where, have you guys ever heard of organophosphates? What is an organophosphate? Usually find it like in certain insecticides. So it used to be used a long time ago. You see it used more in like developing countries. But basically it's a very cheap but very effective insecticide. And actually what that does is it binds to that enzyme, acetylcholinesterase, and actually prevents it from being functional. Okay, so we'll learn when we get to like the autonomic nervous system that when you end up inhibiting acetylcholinesterase, you have too much acetylcholine being around, right? Because now the enzyme's been taken out, and now they end up having this whole toxic syndrome that occurs, where basically they're urinating, they're defecating, they're throwing up, they're tearing up. They have all kinds of different problems that are occurring due to this. So again, you can have disease states where the enzymes are being affected, and disease states where the substrate's being affected as well. And either one, if you don't have both connecting together, nothing's going to happen. Okay. Okay, so yes, ma'am. I can't because of the recording. That'll actually goof up the recording. Okay, is there a specific thing I can answer?
Based on the picture, I didn't understand why the enzymes wouldn't go back to just coming into the body.
It's just really showing how they're just coming together, not really anything to do with the specific actual function of it.
So does it matter like which way it comes in as long as
So based on the actual structure of the protein, again, if you look at like 3D structure of proteins, it looks like a big blob of nothing, right? If you look at some of those Wikipedia ones. But yeah, there will be a certain conformation that the enzyme and the substrate or whatever will come together to form that. So again, they can rotate in a 3D space and kind of come together in whatever conformation but usually there's one receptor site that the substrate will bind to. Yep. Oh, and a good question came up during the break, asking about testing material, right? Because that's what you guys are really concerned with, right? Okay, nervous chuckles aside. Yeah, so you worry about what's going to be on the test. Basically, I try to make things, this is your guys' first semester, so there tends to be a higher preponderance of questions more on regurgitation of memorized content, right? Obviously, at the time where you leave your didactic year, that is not going to be the case. We really try to focus a lot more on clinical-based scenarios to have you guys really think hard, like think critically to come up with an answer. So there will be some of those questions where it's just basic regurgitation of information. There'll be some of those. I'm going to try to also put in some clinical cases as well to try to help make you guys jump through a few mental hoops to get to an actual solution, right? So I'm not going to have you memorize every single example that I mentioned. So is an organophosphate going to come up on the test? Probably not, right? But if I said, hey, there's a disease state where this enzyme is being inhibited that normally causes this reaction, what would be the downstream effect, right? And so by using the information in the question, you can be able to make that kind of mental leap in logic to get to the right answer, okay? So I'm usually pretty straightforward. I don't like to do trick questions, but I do like to make you guys think, okay? So don't be intimidated by that. There will be some of the easy questions, but I try to incorporate some clinical stuff, even though you guys really aren't fully versed in all the clinical world. That makes sense? Hopefully. I'm just babbling at this point. Okay. So anywho, we mentioned that increases in temperature can increase the rates of reactions, especially within a certain threshold. So that's why we like sometimes in fever, that's a good thing by increasing that the temperature of the body. Sometimes you're making those reactions happen a little bit more easily, a little bit more efficiently, but that only is going to work up to a certain case. So if I had a patient who had, say, a fever up to 107, 108 after doing like amphetamines, those enzymes start to denature, right? Sometimes it's a reversible process. Sometimes if you cool the patient back down, those enzymes come back into conformation and can work again. Sometimes not. Just depends. So that's why we like to keep them in the sweet spot for temperature, not let them get outside of that. Okay, and again, based on the pH, this is kind of an interesting example where we're looking at 3 different enzymes. So here you have pepsin, you have salivary amylase, and then you have trypsin. So is anyone familiar with pepsin? Where do you find that at? Yeah, it's found in the stomach. Yeah, so it's helpful for helping to start digesting some of the food coming in through the GI tract. You also have salivary amylase. What does that do? Yeah, it breaks down carbohydrates. Why if you ever like leave bread in your mouth for a long time, it starts to get sweet, right? Because your salivary glands are already sending out amylase and breaking down those sugars into carbohydrates and simple sugars. And then trypsin, what do you think that does? Trypsin usually helps to like kind of break down some proteins and things like that. But trypsin is one of those things that we'll see that they have different ranges of efficacy based on the pH of the environment that they're going to be in, right? So for instance, pepsin, that gets secreted by the stomach. We know that the stomach pH is typically pretty low because you're sending out those stomach acids. And so that's why you see the activity here of pepsin gets to be very high at around a pH of 2. And then as you start to get to more basic environments, start to get more neutral, start to see that start to go down, right? So then by the time you get to the small intestine, pepsin is not really all that effective, okay? On the other hand, you have things like salivary amylase. Again, in the mouth, pretty neutral pH for the most part. That tends to have its peak activity around 6.8 or so, okay? Once it gets down to the stomach, not as effective. And then on the other hand, we have trypsin, which is going to be more effective in a basic environment. So again, sending out from the pancreas into the small intestine where you've neutralized a lot of those stomach acids. Now you have its peak activity here happening between like 8 and 10, right? So just know that enzymes are going to work better at certain environments, certain pHs. This is actually important. We mentioned, I said those cystic fibrosis patients, I mentioned their pancreas oftentimes gets plugged up with this really kind of thick mucus, and so they can't send out things like amylase, protease, lipase. And so what we have to do is give it to them orally. Okay, so what we actually have to do with those capsules is they come in these little tiny beads, and actually what those beads do is protect it from acids. Because if we send those enzymes and they got into the stomach acid, they just get broken apart. It wouldn't be any good for the patient. But by protecting them in this little pH-coated capsule, you're able to send it through the GI tract, it gets past the stomach into a more basic environment. That coating melts off of it in the basic pH because it's more soluble there, and then now the enzymes can actually work on the food that's been ingested. That's one of those cases where you can utilize different pHs and try to get the drug to the specific site we want it to work, right? So just kind of an interesting little dosage form thing that we'll do with our drugs sometimes. Does that make sense? Let's try that right. Okay. So we have lots of different enzymes. You can see varieties of them. The GI tract utilizes a lot of enzymes, both from the pancreas. I mentioned that's kind of its exocrine function when it's sending out things like protease and amylase. The stomach, salivary glands are all very important for digesting the food coming in, breaking down for more complex structures down into more kind of simple amino acids, sugars, things like that that we can utilize and can be absorbed more readily. Because again, one of the things we'll look at when you're looking at absorption of nutrients from the GI tract, do you think smaller things or bigger things get absorbed more easily? Smaller things, yeah, it just makes sense that smaller things will be absorbed more easily. So if we can break it down to as small a molecule as we can, it's going to aid in absorption as well, right? That's what happens with CF patients, so they don't have those enzymes. The food stays in kind of too complex of a molecule and it can't be absorbed very well. So that's one of the problems they run into. Okay, so in some cases you're going to find that there are other things that help these enzymes to work more effectively, or work at all. And so we'll run into coenzymes occasionally. You're going to find that these are small molecules that help to aid an enzyme to work in that reaction. So you can find normally these are going to be organic molecules that are derived from some of our water-soluble vitamins. So say, for instance, like your B vitamins are going to be a big one, where we'll run into like FADH and NADH more commonly when we talk about energy production, but those actually come about from some of your B vitamins. Things like riboflavins for B2, niacin for B3, and that helps to contribute as a coenzyme to help make the NADH and FADH2 actually work in the process for producing ATP, as we'll get into in a few lectures. And then sometimes we'll have cofactors, which are kind of a necessary component to help the enzyme actually work more effectively. So say, for instance, you were to have this Pac-Man looking apoenzyme that will be combining with this cofactor that will actually allow the process to actually occur. So some of these catalyzed reactions require things like calcium, magnesium, even like things like manganese in order to make their actual reactions work. Okay, so this would be called a cofactor that's needed. If it's not there, the enzyme's not going to work as effectively or at all in some cases. Again, just giving you an example of how some of these cofactors are going to be working here. We utilize cofactors quite frequently in the tox world when we have patients who ingest either accidentally or on purpose things like radiator fluid, right? Does anyone know what's in radiator fluid? Yeah, you might find a small amount of that. That's actually called ethylene glycol. It's actually an alcohol. Sometimes people do it. It's an alternative. If they're an alcoholic, they can't get normal, alcohol. They'll drink that sometimes, or if the kid gets into it. But one of the things it does is break down these really nasty organic acids, and it kind of overwhelms the body's ability to metabolize those. And it's not because of the fact that we run out of enzymes, it's because we run out of cofactors, right? And so in those cases, we actually give things like thiamine or folic acid or pyridoxine, which are all water-soluble vitamins, as cofactors to help establish those reactions again and detoxify the alcohol, right? So it's one of those cases where, in some cases, the enzymes need those cofactors. If they're not there, the reaction may just not occur. Yes, ma'am. No, so this would be one enzyme here. I'm going to use a pointer. So this would be one enzyme here, right? And these would be the two substrates that are either, so in this case it would be like catalyzing a reaction where they produce one new product, right? So here the enzyme, the cofactor can come in and help facilitate this reaction, so that way it actually occurs in the first place, right? Make sense? The second picture, this would be another case, just a different confirmation where the cofactor is working just in a different site, yeah, basically. Well, these would be actually two different enzymes here. You can notice the conformation is a little bit different, but it may be working. But notice the substrates are the same. So maybe in one case where you may use different cofactors to help different enzymes work effectively to produce the same product, right? Because sometimes you have multiple enzymes that can do the same reaction. It just depends on how the process occurs. So, for instance, in some cases you may exhaust one pathway for metabolism and actually shuttle it into a different one that still may require the same cofactor but maybe a different enzyme. So I'll talk about that in just a few minutes.
Are cofactors required or do they just help?
In some cases they are going to be required. Sometimes it just may slow down the reaction somewhat, right? So say for instance there's a good example when you are breaking down certain clotting factors. Right, so I think you guys remember like that clotting cascade I showed you a picture of. One of the ways we can break that down is with the enzyme heparin. Okay, heparin can actually be utilized as a cofactor to help a product called antithrombin 3 break down those clotting factors. So basically, clotting factor adds with antithrombin 3 and catalyzes the reaction to break down the clotting factor. So that way you don't have too many clots, right? By having heparin come in, it can facilitate that reaction as a cofactor and have that reaction happen a lot faster. It happens on its own, but heparin helps to make it work a little bit better. So again, sometimes necessary, sometimes not. It just depends on the enzyme. As in medicine, all things are sometimes, maybe, of course it's not black and white.
So it will only help increase the speed of the reaction at cofactor level?
Mm-hmm. Or sometimes allow the actual reaction to occur in the first place.
It
So
will never slow it down.
It'll never slow it down. No. Yeah, only help it work a little bit better. At least I'm not aware of a situation where that would occur. That could be out there, but I don't know it. Okay. So again, looking at the substrate concentration. So as the substrate increases in concentration, the reaction is also going to occur in an increasing rate as well, right? So oftentimes, for a given reaction, you usually have more enzymes available than you have substrate, okay? To a certain point, right? As you start to increase substrates higher and higher and higher, eventually you're going to exhaust the ability for those enzymes to continue metabolizing because they're all kind of bound up, right? And so at that point, you're going to be having this kind of rate-limited metabolism or enzyme reaction because of the fact that all your enzymes are saturated, okay? So a good example of this would be, we have a drug called phenytoin. Has anyone ever heard of that drug? Use it for seizures in a lot of cases. And so this is a drug that normally when your body is, when you give normal therapeutic doses of it, your body can metabolize it just fine, you know, and you never really saturate those enzymes, okay? And so you can achieve, you know, decent levels. Patient doesn't have seizures, they don't have any side effects, and they're good to go. When you take too much of that drug, though, you end up exhausting those enzymes because they're all saturated. They're all trying to metabolize as fast as they can, but there's too much drug around, and then the levels skyrocket, and they have all kinds of side effects from it. So again, all these enzymes can be saturable. It's just a matter of, you know, does it happen on a routine basis, or are we putting the patient into a disease state where they have too much to saturate those enzymes? Okay, so just know that there's always going to be kind of a plateau effect that occurs here, where as you increase that substrate concentration, you can wear out those enzymes, and there's just too much for it to do, and so it's going to be a much slower process at that point, right?
So typically there's more enzyme.
Typically. Yeah, typically you don't get to a point where you're saturating those enzymes unless you were doing something where you're just giving too much substrate, or the patient is producing too much substrate, whatever it happens to be. Yeah, so lots of different example like disease states where that can occur. Yep. And we'll talk about metabolism like that a little bit when we get to the pharmacodynamics section as well. So that'll certainly come up again. Okay. And so again, just note here, if you were to add any more substrate once you get to this point, you're not going to increase that rate of reaction, right? Because your enzymes are all saturated. You hit a plateau effect here. Yeah. Okay, again, just another example showing you the saturation that can occur. Normally, as you add more substrate, the enzymes can react to that. You have enough enzymes to do catalyze that reaction even faster. But again, everything saturates at a certain point, right? You can overwhelm any kind of system. Okay, some of the things you can actually measure enzymes levels in the blood to help you either monitor therapy or to diagnose disease. So here's some example ones that we can actually measure in the bloodstream to determine if someone has appropriate levels, too much or too little, of different enzymes. So some of the ones we look at specifically for things like liver function, we can look at things like alkaline phosphatase. So you ever hear someone talk about ALK-Phos, that's usually just a kind of a shortened way to talk about that. That's usually going to be listed on a liver function test if you ever have a lab value being done there. But other things like we can look for creatine kinase. This is really important for looking at things like if you have muscle breakdown. So if you had a patient who had a heart attack and they're having hypoxia, not enough oxygen to the heart cells, those cells start to necrose and they start to release that enzyme. And so by measuring that, we can say, hey, yeah, you're actually having myocardial infarction, right? That's one way we can do that. Transaminases are another good liver one. So if you're here at AST, ALT, those are ones we're also looking at to determine how well the liver's functioning, right? Normally, ALT and AST should be kept in the liver, right? But if you have that starting to spill out, that probably gives you an idea that, hey, maybe the liver's not, there's some kind of process where the cells of the liver are being damaged and they're releasing a lot of these LFTs. Okay.
AST and ALTs relate to myocardial infarction.
Those typically don't. It just depends on if there's significant enough hypoxia occurring. So say, for instance, you had someone who was like a drowning patient or something like that, right? And so they don't have enough oxygen getting to the tissues because they can't breathe, their lungs are full of water. You can see that you have kind of global hypoxia that's occurring there that can affect lots of different tissues. So sometimes you'll see them kind of in tandem. It's not usually you'll see that like LFTs are a specific sign for myocardial infarction. It just depends.
Right.
Yep.
So it's not like the cardiac enzymes that they test for in the blood.
Yeah, so the creatine kinase is a specific enzyme we'll look for, but you can also look for things like troponins, which are just another component of those myocytes, but not a specific enzyme itself. Yep.
Not
So-
the LDH and the AST.
No, yeah, just the creatine kinase is the primary one there. But you can also, like, we have a lot of patients over at Nemours at the Children's Hospital. Patients will have like mitochondrial dysfunction. Either they are missing a gene or they have a mutation in their genes where they actually don't produce enough of certain enzymes. And so we can actually measure those as well to diagnose mitochondrial dysfunction. And so that's another common one where you don't see those labs done very frequently because usually people get diagnosed pretty early, but that's where we use a lot of kind of weird kind of tests that you don't normally do. But things like CKs, things like AST, ALT, those are kind of routine kind of labs that you'll do, especially like in emergency medicine setting or in the primary care setting.
Could, sorry, could
Yeah.
CPK be increased as a after like intense exercise? Or
Mhm.
something? Okay, because I've seen it like in the ER before. Like,
Yeah. So do you guys know what you call that when you have a lot of muscle breakdown after like a vigorous exercise? Yeah, rhabdomyolysis. So that's another case where you have a lot of muscle breakdown happening, and you can have a lot of that CPK or creatine phosphokinase being released in these muscle cells, and that's how we can measure that and say, hey, they're having a lot of muscle breakdown. We need to hydrate them well and do whatever we need to do. You see that a lot in Florida, especially around this time of year, because you have a lot of those boot camps that are being done. So like you think about Saturday mornings, someone who's not really physically fit goes out and works out for 6 hours straight, doesn't drink enough, rhabdo, like it's going to happen. How does the urine look when you have rhabdo? Looks like tea. Actually gets a very brown, kind of muddy brown colored. So if you ever hear someone talk about like tea colored urine, it's a bad sign. They need to get to the hospital like right away. I also see a lot of pictures like that on the internet or something. They're like, hey, is this normal? And they're like, no, you need to go to the ER like right now. Like, oh, I'll just wait it out and see. I'll just drink a lot of water. Like, nope, your kidneys are going to shut down. That's not good. Yeah, so that can be a big thing you'll see, especially here in Florida, being so hot and having people are not physically fit, kind of trying to overdo themselves.
thank you.
Yep. So talking about metabolic pathways, so we can see that a lot of reactions occur kind of in a sequential chain over time, and we call this a metabolic pathway. So you will begin with an initial substrate. Here, enzyme 1 will change it to product B, enzyme 2 will go to product C, D, E, and F, and you get your final product, right? We see this a lot with like drug metabolism, where it'll go through multiple steps to get to a place where it can be excreted to the kidneys or to the liver. But normally it's what we call a single pathway for metabolism of a substance. Actually, I'm already over. I'm going to go ahead and cut this. We'll continue with this section. I'll go over the slide again, and we'll continue on next time. But I'll give you guys a break now. We'll come back and then do pharmacodynamics.